Uh-huh.
I have overcooled a sea lion.
In California? Dang. Oh, that's where we're going. Yeah. Yeah, we're sorry. Yeah.
I know he's
We're
talking
doing more of
sounds
that.
getting excited. Okay, hi everyone that's here. Thanks y'all for coming. I know a lot of classes got canceled today, so I still appreciate you guys coming out here today, if you were here already or not. So today I have the pleasure of introducing Dr. Ryan Jones. To be honest with you guys, it's still kind of weird to call him Dr. Ryan Jones.
You still have to.
I apparently still have to. We met about like 4 years ago in Santa Cruz. It was my like kind of first introduction into marine mammals at the Pinniped lab there. And he was just a little wee baby. He was like just in his second year of his PhD. You know, we were a little younger then. We had a little bit more energy. Yet somehow he was still always working so hard and always so dedicated to everything he was doing. So let's see here, all the specifics. Ryan graduated from UC Davis with a Bachelor's of Science in Biology with an emphasis on medicinal microbiology. If you know him now, that makes no sense to what he does now. So only one short year later, he was at the National Marine Mammals Foundation working with sea lions and dolphins. During that time, it seems like he worked on like a dozen publications or something. I was looking through. That was shocking to me. And that was right before beginning his PhD at Santa Cruz in that Pinniped Cognition and Sensory Systems lab. This is where he met the true love of his life. She is sweet, a little sassy, really beautiful sea lion named Ronan that he just adores, and she adores him. So I'm sure you guys will see some photos later if he chooses to share those with you guys. Anyways, he's been busy since these times. He's led a team in Antarctica with plans to return again this winter, I believe. Nice. Is a research and program manager for the California Ocean Alliance, and most recently began his postdoc at the UCSC Costa Lab, exploring the effects of anthropogenic noise on California sea lions. Because like I told you, he loved Ronan very much. So anyways, please give me a hand in introducing Dr. Ryan Jones.
Okay. Thanks, Sebastian. That was so nice. Okay, I'm going to start a timer because I was told to keep it relatively short. And when I was invited to come do this talk with you guys, I kind of went back and forth on what exactly it is that I should talk about. I had this whole thing where I was like, oh, I'll just talk about all my dissertation research, or I can talk about the work that I'm doing now, which I'll cover a little bit at the end. But then I kind of figured instead of doing all of that, we'll just talk about a concept that I have really fallen in love with in terms of understanding auditory biology, which is auditory masking. And we're just going to kind of do a nice deep dive into auditory masking and how it works, and some of the specifics on what I was interested in studying during my PhD, and how we can kind of take the data that I collected and the research that I did with these captive animals at Long Marine Lab, and how we can take that data and apply it to understanding how anthropogenic noise, and in this case, more specifically Navy sonar, can impact the animals that we're interested in studying. Luckily, I can skip my about me because Sebastian really covered it really well, so we're just going to blow right through that. And then before I get started, since there's not that many of you here, I can kind of get to know you guys a little bit. And is anybody working with acoustic data at all? So yeah, a little bit. What are you working on?
GPS arrays using acoustic transmitters.
Nice. Okay, perfect. That kind of stuff it applies to this a little bit, but we're thinking more about like auditory reception in terms of hearing biology. But if you're doing anything that's putting sound into the ocean, it's important to consider these types of concepts. And then is anybody working with like sea lion data at all? Okay, perfect. We're just going to introduce you guys to all the things I like, which is perfect. And to kind of bring us back, this was a picture that was taken on Año Nuevo Island, just up the coast a little bit, where we do a lot of our work with sea lions. This is a Steller sea lion pup and his mom in the water, and they're calling back and forth to each other. In this case, I think she was kind of telling him to get in the water, and he was fighting her on it a little bit. And this is one of the kind of many interactions that you can see between conspecific, in this case, sea lions, which is kind of going to be the majority of what I'm focused on for this talk because it kind of brings us together. But the types of interactions that you see between these types of marine mammals that are based entirely on acoustic signals. So just a little overview of sea lions. They're amphibious carnivores, which means they're spending a good chunk of their time hauled out on land, but then they're also spending a really good chunk of their time swimming around in the water. And like every species of marine mammal, this is like one of the very few things that I can feel comfortable in saying definitively, but all species of marine mammal in some capacity are reliant on acoustic information, right? So does anybody, well, I guess I won't make you guys answer questions. That's usually my go-to. But the reasoning for that is if you think about the marine mammal system, right? It's this terrestrial animal that you're then it's like taking a dog and dunking it underwater, right? So now all of these systems that they've developed to work and function well in air now have to function underwater. And that kind of precludes a lot of types of sensory information that they have access to, right? I mean, one thing we know for sure is that there's not that much light in the water, right? It's pretty hard to see, especially if it's really turbid. So light and vision isn't really a great option. Feeling is a good one, right? They have really sensitive pinnipeds specifically, which is our sea lions, seals, and walruses. They have really sensitive whiskers or vibrissae on their face that they can use to track prey from relatively good distances, like up to 10 meters or so. But that's not really good for any sort of long-range information, right? And then the one thing that works better in water than it does in air is acoustic information, right? So acoustic information travels more efficiently underwater than it does in air, and it will travel farther underwater than it does in air. So overall, all marine mammals are reliant on acoustics in some capacity. Sea lions particularly are really reliant on like mother-pup contact calls, which is what we see kind of in that picture we were looking at earlier. Male-male competition, a lot of times the males will display to each other using vocal calls, and they can pick up on like killer whale echolocation signals to help avoid predation. All of which is to say is understanding the acoustic biology or the auditory biology of sea lions and marine mammals in general is extremely important. So if we want to be able to understand how these animals are functioning in their environment, we have to have a pretty solid understanding of their auditory biology. And one of the main tools that we use to understand auditory biology is this little guy here. So this is what we call an audiogram. And what it is just a graphical representation of what your subject, in this case a sea lion, what types of acoustic signals they can hear. So I'm going to orient you to the plot a little bit, and then we'll talk a little bit more about it. So on the x-axis, we have frequency or pitch, right? So that's increasing across the x-axis. And on the y-axis, we have the animal's hearing threshold. So it's basically like level, right? So how loud of a signal it is. So what this plot is showing us is the threshold level, or the lowest signal that that animal can hear at a given frequency. So these are usually measured with like pure tone, so like a really kind of clean signal. So in this example for a sea lion, this was a audiogram that was measured in air. You can say at 1 kilohertz, well, we can go maybe 4 kilohertz or so, which is where our voice, our speech usually is. The lowest signal that that animal can hear is at about 10 or 15 dB. Does that make sense? So when you're looking at this plot, everything above this line is what we would consider to be audible to that subject, right? And this is, again, a sea lion in air, but because they're amphibious, so if this isn't something you would have to do with, say, a dolphin, but because they're amphibious, we can measure an audiogram for a sea lion in air and also underwater. It looks a little different, right? We'll talk a little bit why that is. They're a little bit more sensitive. Again, the lower your threshold value is, the better your hearing is at a given frequency, right? They're a little bit more sensitive in water over a wider range. But the most important thing about these audiograms specifically, and when we measure audiograms in general, is that these were measured in perfectly quiet conditions with trained animals, right? So this is a big chunk of the type of work that I did during and before my PhD was where we administer hearing tests to trained animals. So it can be done in a variety of different ways, again, both in air, like this in the acoustic testing chamber that we have at Long Marine Lab. That's Ronan in there, hard at work. Her whole dissertation on her back. And then that's Ronan again doing the same type of hearing test in water. And basically the way it works I'm going to fall off this little stage. If she rests her little chin in that chin station, we'll play a signal, like a pure tone. When she hears it, she pushes this little paddle over to her left. And then we make that tone quieter and quieter and quieter until she stops responding, right? You guys taken hearing tests recently? It's the same exact thing. And every time she hears that tone correctly and pushes the paddle, she gets a little fish, a reward to let her know that she was correct. Every once in a while, too, we'll just have her sit there and play no signal, make sure she's cheating. And if she waits on the chin station, then she gets rewarded again. Okay, so the whole point of doing these measurements, though, is that we want to understand, again, remember, like, what is the quietest signal that she can hear in her environment, right? So we go through all of this effort, like the pools. I mean, Sebastian's had to do it a lot. We have to drain the pools a little bit so there's no sloshing. The whole compound gets shut down. This room is like dead quiet inside, so you can get to the actual limit of hearing for these guys. But the big thing to consider when you think about, again, sea lions in particular, is that I don't know how many times you guys have spent around. I mean, there's sea lions all the way. It's nice. You guys hang out with them all the time. But they're really loud, right? And so audio is great on this. It's a little crackly, right? So it's very, very rare that you're going to have a sea lion that's in an environment that's this quiet, right? And I think the main takeaway there is that these absolute measures of hearing, which is what we would call the types of measurements that we take if we're collecting an audiogram, aren't normally representative of what an animal's in situ hearing capabilities are, right? It's just not logical to think that they're often going to be in an environment where it's dead quiet. The only times for, especially pinnipeds, that I would think that this does apply is in the poles, right? Where it's very, like, there's a lot of ice cover, there's not as much like wave action or wind, they're not in these big colonies. A lot of times these audiograms are pretty applicable, but for a species like a sea lion or other animals that are in kind of big social aggregations, these measures of absolute sensitivity don't really tell you too much about what kind of what types of auditory information they actually have access to in their environment. Okay, we're still on board? Sound good? No one's fallen asleep yet? Okay, so that brings us to what we're going to really be kind of focusing on. And I'm going to try my hardest. I don't do a great job when I'm looking through these slides to not show you guys too many like data slides. It's more just to have you kind of have a good understanding of what this concept is. I just want you to walk away having kind of some basic idea of how auditory masking works in the ear. And if you're not that interested in sea lions, it applies to all mammals. So it can apply to yourself a little bit. You can kind of just think about it happening inside of your head if that's the only thing you want to do. But the only thing that I really want you to walk away from today is just a little bit of an understanding of how this phenomenon works. So auditory masking, kind of baseline definition, is when one acoustic signal interferes with the detection of another. And this is something we experience all the time, right? If you're in a bar and you're trying to have a conversation with somebody, but it's too loud, there's too much other noise going on, and you can't hear what they're saying. You're being masked by the noise that's going around you in the bar or restaurant or playground, wherever you want to hang out. And auditory masking is what we consider to be a cochlear phenomenon, and I'm going to go into this in great detail. I love the cochlea. So we're going to kind of really break that down on kind of where auditory masking is occurring in terms of your sound reception pathway. And the primary metrics that we use to describe auditory masking are critical ratios and critical bandwidths. And again, we're going to go through the definitions of those two terms too and look at a little bit how we can apply, how we can measure critical ratios and critical bandwidths in trained subjects, and how we can use that data to get an understanding of what kind of an animal's auditory environment looks like. Okay, let's start with a little bit of discussion about the cochlea, but we have to get there first. So this is a kind of a representation of a kind of your standard mammalian ear, right? So they all kind of work in generally the same way. Your auditory meatus, or your kind of external ear, is right here, right? And in any sort of hearing scenario for us in air, I don't have my little arrows, so I'm going to have to point. To receive an acoustic signal, as acoustic energy out here gets funneled through your auditory canal, this tube that you can kind of stick your pinky in a little bit, your auditory canal, and it stimulates the tympanic membrane. It's like this kind of stretchy trampoline that's at the opening of your auditory canal here. And it stimulates the tympanic membrane, which vibrates the 3 ossicular bones in your ear. These are your ear bones. And what those do is they act as amplifiers, right? So they take that information, that vibration from the tympanic membrane, and they amplify it before it gets sent through the oval window to your cochlea, right? And he's the star of the show. He's who we're going to really be talking about. But something to keep in mind, too, when we think about an amphibious ear for like a sea lion, this is all air, right? The acoustic information, air, fluid, right? So that's why you need these bones here. Those ossicular bones are amplifying the signal before it goes into the fluid-filled component of your inner ear, right? And it's because you lose a lot of acoustic energy when you transfer between mediums. Think about like if you're at the pool when you were a kid, you didn't want to get out, your mom's telling you to get out, you just dunk your head underwater, can't hear it anymore, right? It's because that acoustic information has a hard time going from air to water. Same thing happens inside of your head, which is why you have these bones to kind of counteract that loss of, it's called impedance mismatch, but that loss of energy. Interestingly, if you take this ear, it works the same way for a sea lion, right? Dunk it underwater. Now you're going from water to air to fluid, and it gets all messy. And that's why the auditory sensitivity plots are a little bit different between air and water. It's because they're using a different way. They're not reliant on this kind of normal pathway, but they're just taking all of their fluid-filled fascia in their body, using that to funnel the acoustic information just straight to the cochlea, right? So they kind of bypass the normal auditory pathway. And that's why they're a little bit more sensitive in water, because they don't have to rely on that kind of standardized way. But the important thing is that all that acoustic information gets focused into the cochlea, the little snail on either side of your head. Now, the cochlea is fluid-filled, again, which we covered in pretty great detail, and it's frequency-coded. And now what that means, we can take a little snail and we can unroll him. It doesn't hurt, I promise. We can unroll him, and then what we can see is that it's divided into these little sections that are frequency-coded. So like a given frequency is going to be coded by the basilar membrane in a little area. And I'll get into this a little bit more. But the way that the membrane works, it's like a tarp, right? And it's sitting on top of your little hair cells. So if an acoustic signal comes in at a given frequency, so like the tautness of this membrane is coded to respond to a given frequency, if it moves, it stimulates those hair cells underneath, and that sends the information to the brain that lets you know that you heard something. So this frequency coding is really, really important when we think about how the ear works and how it's kind of taking acoustic information and divvying it up and coding it or transferring that information to the brain. And we can think about how the basilar membrane or the cochlea is frequency coded as a series of overlapping bandpass filters. So a bandpass filter just means that it has an upper and lower cutoff frequency. And so like these numbers don't mean anything. But let's just say that this portion of the membrane is coded to trigger between 1 and 2 kilohertz, right? So if a 1.5 kilohertz signal comes through the ear, doesn't move, wiggles a little bit, doesn't move. The wiggle here lets your brain know, oh, I heard a signal at that given frequency. We good there? A lot. I mean, especially if you're not an acoustician, it's a little tricky, but I promise, as long as I explain it to you 10 more times, it'll start making so much sense. And the way we describe kind of the way that these overlapping bandpass filters work are those two primary metrics of auditory masking, right? Because it has a lot to do with what types of signals are interfering with each other. If you're dealing with information that's all within the same frequency band, they're going to compete with each other for attention, right? And that's how you kind of block out certain pieces of information, is because there's too much going on in that little band. So we can describe them in two different ways, the critical ratio and the critical bandwidth. And I'm going to try my hardest to take this super slow so it makes a little bit sense, because then when we start looking at actually what the measurements look like, it'll make a lot more sense if we have this background, right? So let's just say, again, this is the same kind of like graphical representation that we were looking at with the audiogram. You have the level or loudness of your signal on the y-axis and the frequency or pitch of your signal on the x-axis. And this is just a representation of the ambient noise in your test environment, right? So, at whatever frequency this is, the noise in the surrounding background is this loud, right? And so let's say we put our little sea lion in the mix. We give her a hearing test at a given frequency. Her threshold at that one frequency is whatever this value is. We can just say it's 10 dB or something. Doesn't matter. It's all relative. Right? So that's her absolute threshold. It's not influenced by the background noise at all. She's in a perfectly quiet environment. That's the quietest signal that she can hear at that given frequency. The next thing that we'll do is we'll introduce noise into the background, right? So we'll play like a little section of white noise, like, and give her the hearing test again. What do you guys think is going to happen to that threshold? Think it's going to go down or up? Sebastian's cheating because he helped me collect a lot of this data. It goes up, right? Because her hearing is worse. That makes sense. In a noisier environment, the quietest signal that you're going to be able to pick out is going to be louder, right? So that just kind of intuitively makes sense. Threshold's going to go up, and that's her masked threshold, right? And the cool thing is that amount by which that threshold goes up is your critical ratio. Another way to describe it is that it's the signal-to-noise ratio required for the detection of a signal in noise. So it's a very standardized number, and I'll get into how that number changes based on frequency. But if you were to increase the noise level, the threshold still goes up, but it goes up, again, by a critical ratio above the noise. And I think you can go down. And then what's happening next? Yes, okay. Hold on, I think spoiler alert, but this happens. So let's just say if we take in the frequency content of that noise, right? So the bandwidth is what we call it, where that noise frequency starts and where it ends. If we take that and we make it sufficiently narrow, so it's like, goes from, if this is a 4 kilohertz tone, maybe it goes from like 3.5 to 4.2 or something like that. It's very narrow. You'll get what we call full masking release. So that's like that noise isn't even there, right? And then if you increase the bandwidth of the noise, little by little, eventually the threshold will climb a little bit, right? It comes a little higher as you get wider, but then essentially you can make it as wide as you want and the threshold doesn't change at all. So that point at which you can extend the bandwidth of your noise stimulus and see no associated increase with hearing threshold is what we call the critical bandwidth. It's a little tricky. It'll make a little bit more sense. But that bandwidth, the critical bandwidth, is analogous to the width of that bandpass filter that's associated with the cochlea, right? So the frequency coding within the cochlea, that width there is basically analogous to your critical bandwidth. Is that tracking? So we have a critical ratio, which is how loud the signal has to be above the noise for it to be detected by your subject. And you have the critical bandwidth, which is the frequency content of the noise that's contributing to the masking of a signal. I think I spell it out a little bit cleaner here. Yeah. No, actually, I basically said exactly that. Perfect. Good for me. Right. So signal-to-noise ratio required for detection and the frequency band of noise. And so let's just say all of that we were talking about was at 4 kilohertz. That only applies to that subject at 4 kilohertz. Those values, the critical ratio and critical bandwidth values, only apply at a given frequency because they're frequency dependent. So they're going to change in a really stereotypical way, which makes things a little convenient, but they're going to change dependent on your frequency. The very cool thing about it is it does not change depending on the medium. So if I were to do a critical ratio measurement for Ronan in the chamber and in the water, they would be the exact same. The thresholds themselves would be different, but the amount by which the threshold has to exceed the noise is going to be the same either way. And the reasoning for that is because this is all occurring downstream of the sound reception pathway. So it's all happening in the cochlea. All of the processing in terms of receiving the sound has already been done. Oh boy, this next part, I've never tried to explain it out loud before, so it's nice that you guys are a captive audience, but the way that this works, let's just have a little bit of a representation of our auditory filter, right? And again, this is kind of what I was getting at in terms of the competition between noise and the signal that the animal is looking for, right? So let's just say, again, this is just a representation of the critical bandwidth. So this is like frequency on the y-axis here, or on the x-axis there. And again, that lines up with the coding on the basilar membrane within the cochlea. And again, frequency on the x-axis, level on the y. And then let's just say that this is a representation of our pure tone threshold, right? So we're trying to think of all the energy within this pure tone that's what's needed to register to trigger the basilar membrane into kind of stimulating those hair cells, right? If we introduce noise that's sufficiently quiet, it's not competing. So the energy, again, you're only looking at the energy within this bound here, but that's not enough to change the amount of energy that's in that pure tone. That's still enough to stimulate the basilar membrane, right? But if you increase it, now it's suddenly everyth's lost. So the resolution inside the basilar membrane isn't enough to trigger it to let it know that you've heard the pure tone. It's all masked. So it has to come up. And the amount that it needs to come up, again, we're only looking within the critical band. But the amount that it's needed to come up, that's sufficient in order for it to trigger the hair cells underneath. That make a little bit more sense, hopefully? Okay, good. But then again, if you make it narrow, you don't need as much, because there's not as much of a competition between the energy in the target signal that you're looking for and the energy that's contained within the noise stimulus that you're listening to. Good. That's the last of my little technical slides that we have to go to. Now we can look at some pictures of walruses if we want. So having a good understanding of what all those are is kind of the first step that you need to do in order to actually measure these in actual animals, right? So you have a good understanding. You want to know what their critical ratios are across frequencies. It's going to change because those, the bandpass filters widen as frequency increases, right? So it's going to be a little bit different in terms of how much noise energy is competing with that pure tone. So you have to do it a bunch of across the different frequencies that the animal can hear, and you have to present them with a bunch of different noise stimuli. So for part of my dissertation, I did these measurements with trained walruses down at SeaWorld San Diego, and this is what the tests look like. There's sound for this one too. Can't really hear it. So basically, instead of sitting in a chin cup, she waits on this white paddle. Here she just gets rewarded for sitting and waiting. It's a little sped up. Sits and waits. Beep. Touches the paddle. And that's how she knows that, yeah, and you can't hear anything out of that. But that's what the masking noise sounds like, so that's pretty good. And what you do is you do these measurements again and again and again. It takes a couple of weeks at a couple of different frequencies. And you can measure those actual critical ratios. So these aren't hearing thresholds that we were looking at before. These are the critical ratios. So again, as you increase in frequency, the signal needs to be higher above the noise to be detected, right? You go down in frequency, the signal can be a little bit closer to the noise. And again, that has everything to do with the shape of those auditory filters, right? Lower frequencies, they're nice and narrow, which means that the signal doesn't have as much noise content to compete with. As you get higher, just a little increments in noise are going to increase your threshold by quite a bit. And I have critical ratios plotted here for sea lions and walruses. Something that I hope jumps out at you is that they're very similar. In mammals in general, you can kind of throw them all together on a single plot, and they're all doing the exact same thing. They're basically increasing by 3 dB per octave as you go up. It just depends on the species. You might have much lower critical ratios like you would see in phosid seals. The phosid seals are really kind of specialized in their hearing capabilities. But in general, mammalian critical ratios behave very similarly. It's just important that you take these measurements with as many different species as possible to confirm that that's what we're looking at. Now, in terms of critical bandwidth, do you think that critical bandwidth is going to increase with frequency or decrease? Who thinks increase? Decrease? Let's look at this, right? So the critical bandwidth, remember, is analogous to the width of your bandpass filter or your auditory filter, right? So they should, in theory, be getting wider, larger as you go up in frequency, which is exactly what we saw in all 3 species of pinnipeds. So walruses, sea lions, and seals, generally, as your center frequency increases, the value of that critical band also increases, which totally tracks with how we think that those auditory filters are working. Okay, that tracking? Great. Those are the only real data slides that I think I show. But let's think about how, now we have all this information, right? We have learned how critical ratios and critical bandwidths work. We know why they work, and we've measured them in a couple of different species. How does that help us? Exactly, right? So what we can do, and this was kind of a way to back check our data as well, is we can use them to kind of predict what we think hearing in a noisy environment would actually look like. So we measured these critical ratios. This is going to be an example using the walrus data. And this is our ambient noise. Remember that little squiggly line from before? So this is just the ambient noise in the test enclosure that we were testing the walruses in. We measured their critical ratios and added that to the noise value, right? Because in theory, if we were to measure their hearing in just regular noisy conditions, their hearing thresholds should be 1 critical ratio above the noise, right? So we can add that little dotted line in there. All this is whatever noise value this is, critical ratio added to that. That's our theoretical lowest threshold. They shouldn't be able to hear or detect signals that are below that line. And then we brought them out. We didn't play any masking noise in the background, and we tested their hearing in that environment. And this is what happened. Landed right on top of that line. Pretty nice, right? So now we can say you can take a noise measurement. We have these critical ratio values that, in theory, they were the same for 2 individuals. So, in marine mammal work, that's good enough for every walrus. And you can take measurements out in the environment and be like, okay, what is their actual ambient hearing threshold going to be like in their environment? You can take a noise measurement, add their critical ratio to it, and that's generally where their threshold is going to fall. What do you guys think is going? Is this a little laser point? Yeah, it's going on here. It's way above the line. Any thoughts? If it had been way below the line, I would have been pretty worried, right? Because that means that our critical ratio logic isn't really working. But when you have it way above the line like that, all that means is that the ambient noise that the hearing was being measured in has no effect on their hearing. Their hearing threshold is so sufficiently above that noise that it's not impacting their hearing threshold. So that right there, we don't have an actual perfectly quiet in-air audiogram for a walrus because it's hard to get them into kind of like a chamber type situation. But if you were to measure that full audiogram, that point would probably fall right on it because it's just an absolute measure of hearing by virtue of them not being very sensitive at that frequency. Does that make sense? Kind of nice little use. And a lot of the critical ratio or auditory masking data that we've collected in the past, most of this has been done in birds previously. Birds are nice to study because they have a really narrow range of hearing, and you don't have to test as many frequencies as you would with a different mammal. But what we've learned a lot about in terms of critical ratio or auditory masking in birds, we've been able to then apply to our studies of marine mammals. And the other thing to think about too is that, birds are in very noisy environments, right? So a lot of what we know about how birds are calling between each other, that kind of informs how we kind of go about, oh gosh, that does not, that looks gross. Okay, very blurry, but we can kind of take the same kind of concept, add those critical ratios to ambient noise environments that we see in songbirds, and we see that their calling kind of lines up with them and you're able to detect it in those noisier environments, right? Okay, that makes sense. But we can bring it back to marine mammals. So something that we've really seen, well, I guess we're not in marine mammals yet, sorry, in terms of critical ratios and critical bandwidths that kind of are against the norm, is that a lot of times for certain species, it behooves them to have super narrow auditory critical bandwidths at given frequencies that they're really, really reliant on. Can you guys think of what types of animals might be reliant on a very specific frequency for life history events that might require them to be really specialized at those frequencies? Yeah.
Deep sea where light's limited.
Light's limited, that's a really good point. You think even more specialization. What reliant on a very specific frequency range? This was one of the questions in my QE. The answer is animals that echolocate, right? So most of the study has been done in horseshoe bats, but we've also seen it in porpoises. Animals that echolocate at very specific frequencies at a very high frequency where their critical band should be really wide, they just have one that's very, very narrow, and that's where they're doing the majority of their echolocation. And what that does is allows them to receive those echolocation returns in kind of like a unmuddled environment, if that makes sense. Okay. And then I think that, I'm almost done here. And just to talk about that, these are measurements that are taken using kind of a lot of conservative measurements in terms of we're using very flat noise. We're in still very controlled acoustic environments. And it's a kind of more of a conservative way to predict how noise is going to impact hearing, right? And this is, again, relatively low-level noise, just kind of the same kind of crowd noise. That has nothing to do with impulsive noise. But there are a couple of things to consider when we think about applying these data to animals that are hearing in nature, one of which is co-modulated masking release. And what that says is that, the masking noise that I showed you is kind of like a square, right? It's very flat on top. That's what it looks like when you record it in like a noise recorder, right? It looks very clean and square. There's nothing going on. Mostly noise is very jagged, right? And what animals can do in that kind of co-modulated noise is do what's called dip listening. But they can sneak and get little bits of information in the breaks and pauses in the noise that kind of naturally happens. So what that will do is that will kind of artificially, or not artificially, but it will decrease those hearing thresholds in noisy environments because they're able to kind of take advantage of little dips and spaces in the noise. That said, you can counteract it. This is just kind of to tell you that we've done this before in dolphins specifically, where we, instead of using like standard noise, we used ice squeaks and ship noise. And we found that, especially for ice squeaks and kind of more inconsistent noise, their thresholds come way down in the presence of that noise, as opposed to standardized white noise. Have that slide in there twice. And then the last point is this idea of informational masking. And this is not something that we have as much of an understanding about, especially when it comes to marine mammals. It's a little bit trickier to study. But the idea, and I can't help, all of my examples have to do with being in a bar, but think about, again, you're in the bar, right, and you're having a conversation with somebody, it's going super, super well, but it's really loud, right, and you can hear that person is talking to you, but it kind of sounds, you can't really, they're talking, you can receive that signal, but you have no idea what they're saying, and assuming you haven't had like 8 beers, then what's happening here is this idea of informational masking, which means you're receiving the signal, but you can't decode it, right? And we assume that this is also happening with animals in the wild as well, where, it's like, sure, in theory, our little guy here can hear his mom. He's probably pretending not to hear her, but because it's so loud, he can just hear that the call is happening, can't necessarily decode the information within that call. And where that gets really important is that the way that these guys reunite and the mom goes out and forages and then comes back to her pup, she'll call out, and then it's really cool. I've just spent a couple of weeks on San Nicolas just waiting for a sea lion wearing a tag to come back. She never did. But I spent a lot of time sitting on the beach, and you can watch these female sea lions coming from the beach, like coming towards the beach. They're calling, and then you see one tiny little baby sea lion go, he pops his little head up, and he crawls, and runs down, because he can identify his mom's call over that kind of wide range, right? The noisier it gets, the harder it's going to be for those animals to kind of distinguish between different types of calls. They may be able to hear that a female is calling, but they're not going to be able to kind of tease apart. It's like hearing somebody yell, but you can't hear exactly what they're saying, right? So this is why this idea of, informational masking would tell us, we would think that the threshold's actually going to be higher in the presence of noise, and then you kind of factor in this idea of co-modulated masking release where noise isn't that consistent, that's actually going to make your thresholds lower, kind of evens out with this kind of approach of measuring hearing thresholds in a very kind of standardized way. And that brings us to this kind of last idea here, where it's auditory masking is just one of the many effects of noise that can impact a marine mammal. It ranges, if you think about a noise source, what it's going to do to a marine mammal. The worst thing that's going to happen is it's going to hurt them, right? It can hurt or kill them in a very physiological way. This is where a lot of those mass straining, we don't know a lot about how those mass strainings works, but there is a physiological response. Sometimes there's a cardiac response that can happen from this kind of impulsive noise exposure. Not something we understand super, super well. And then kind of zoning out from that, you have PTS, which is a permanent threshold shift, which means your hearing is permanently damaged. Those hair cells are kind of rendered null. Scaling up from that, you have TTS, which is a temporary threshold shift. That's like if you're in a concert and you're standing too close to the speaker, and then for a little bit afterwards, you can't really hear anything, and everything's kind of muddled. Animals will experience that in the presence of kind of either impulsive or really long exposures to noise. And then, a little bit different is this idea of behavioral disturbance. That's going to maybe it doesn't hurt them physically or change what they're actually being able to hear, but they're going to change their behavior as a result of hearing that noise. And then the last one is that they can just hear it. It doesn't affect anything, right? So the idea of auditory masking, though, it exists across this entire continuum. It's happening no matter what. Sometimes they can hear it. It's going to affect their hearing thresholds in a way. If it's affecting their actual hair cells and being able to detect signal, that's still masking in its own way. So that's the interesting thing about auditory masking. It's occurring across this entire continuum. And having a good understanding of that kind of helps us inform how we dictate each of these kind of phases of noise exposure. And that's what I'm working on now, is instead of kind of looking at the hearing and the reception component of it, we're looking at that behavioral disturbance component of it. So we're tagging sea lions, exposing them to, well, we will expose them to sonar of a relatively low amplitude and see how that changes their behavior, their foraging behavior. But the most important thing is to have a good understanding if that's a signal that they can actually hear and receive, right? And in a noisy environment, that's a lot of information. Like, you have to make sure that you're playing it loud enough to hit behavioral disturbance and not get TTS, but you also want to make sure that it's kind of fitting into this continuum as well. So it helps really inform those behavioral response studies and also gives you an idea of how they're kind of existing in their environment. Okay, I think that's it. Yeah, that's everything. I'd be more than happy to answer any questions that you guys have, but I also understand if you don't have any. But that's totally fine. I was exactly 40 minutes, so A+ for me. No pressure. I've never given a talk like that in front of non-acousticians, so that, sorry.
This isn't directly related actually to the marine mammals, even though this is super
Totally fine.
interesting.
Yeah.
I'm in the vert lab.
Yeah.
So, this is great. But in terms of when you're chewing something in your mouth, and it could probably apply to marine mammals as well. It's like it's louder. As I was snacking, I was thinking about that.
You weren't listening. You were chewing.
It's
Yeah,
like
that's
it's
fine.
so much louder in your mind.
Uh-huh.
Can you explain how that works scientifically, and is there any relation or studies about that?
Yeah, so that's a really great question. For those of you who didn't hear, it's like this idea of internal noise and how that's going to kind of mask what, it's internal masking. We have, I had a whole section in here about kind of anti-masking strategies, right, that are ways for us to kind of avoid having this happen. It's like moving your head around, but this doesn't really account for your internal masking. And this is something we think a lot about when we're doing the chamber work, because if you've ever, you've been in there, and I'm sure you have as well. You go in there, you can hear your mouth moving a little bit, and it's a little bit distracting too. So it's like, how it's working is really it's your ears are responding to that, right? And it is masking it a little bit. There's like, it's working the exact same way as any sort of noise would. It's just you have better access to it because it's happening right by your ear, and so it's kind of bypassing, again, like I said, that standard auditory pathway, but it's still going right into your cochlea. It's that same bone conduction is what it's called, where it's like funneling all of that information. And it's happening right here in the fluid-filled portion of your mouth and going straight into your cochlea. And that's how that masking is working. But it's the same exact concept. Yeah. But it's hard to measure. That would be the other thing. Is like, how do you, the best way to do it is when you play noise like that, first of all, you don't feed the animals and then immediately give them the hearing test, so them swallow and settle for a second. So you try to remove all of those components, but it is really hard to avoid. Like if I'm sitting in there trying to give myself a test, if you swallow at the wrong time, you might miss the signal when it's really, really quiet.
Great talk. I was curious on how you account for like personality differences of sea lions and walruses and things like that, and how do you know that they're actually food motivated, or what would you do if they weren't food motivated?
Great. I'll say first and foremost that the easiest way to account for all of that is to just do you don't just get like a day where you do your hearing test and you're like, okay, that was pretty good. I think we got it. You do them over months, sometimes years, like every day. Like this is like data that is averaged out to try to account for like their day-to-day motivation, their day-to-day interest in participating. Something that's really interesting and really cool is for animals in these types of situations, they do really seem to enjoy doing the task. So most of the time, they're motivated to participate, and if they're not feeling up to it, then you just don't run that day. For the sea lions, they love it. It's like they're like border collies, they're like ready to go. The walruses, they go through like this weird phase where they're on fire for like 4 days and then they need to sleep for 3 days. And they just sleep in a big pile for a couple of days. And you just have to kind of strike while the iron's hot, while they're feeling motivated. And then, it's like just like if in a classroom full of kids, you don't have they're not all scientists, right? So it's like you have to train subjects over months and months. And at certain points, sometimes you're like, well, this animal just isn't something that they really want to You see that more in dolphins, where it's like they don't want to, they don't have to. So nothing is forced. You don't like constrain diets. That doesn't get you anywhere or anything like that. They still get their allotted diets every day, no matter what. I've been very lucky in my experiences to see the majority of the animals see it as a kind of an enriching task that they get pretty excited. But that's not to say every day is a good day. That's a good question. Anything else?
I got 2 questions.
Oh.
But one of them hopefully is really short. I don't know as much about the aspect you said of them. Oh, sorry, into Zoom. Question 1. How do they hear in water? I don't remember as much about like the fact that they kind of hear through their body. I know like dolphins will do that, right? Like
yeah, and I mean we can do it. We have the capacity to do it too. And it basically is again just this way to bypass that classic auditory pathway. And we know a lot about this, especially in phosid seals, but they have extra cavernous tissue in their auditory canals that when they dive, they fill with blood. Right, so it kind of just almost entirely occludes that auditory canal. The thing for most of those is that that's probably just because it's helpful for diving to exclude those air spaces, but it also has this nice little neat side effect of really helping them receive acoustic information as well. So, and basically all that does is it's the fluid surrounding their bodies, that acoustic signal travels really well from fluid to fluid, so it just transfers really, really easily up to the cochlea. It makes it really hard to localize though.
I think I thought of that blood filling as like that's their way of hearing in water. And
It helps.
It helps. Yeah,
helps.
exactly.
It just removes that air-filled cavity. So.
My second one's a little bit more technical because I don't remember this as much. But you were talking about the masking noise and how their critical ratio, whatever their lowest possible hearing is always going to be 1 critical ratio above what the ambient or masking noise is, right?
If the ambient noise is sufficiently loud. If it's too quiet at a given frequency, then it's back to being based on their biology.
Okay, so that's kind of I think what I was going to ask is like what if the masking noise is like a few dB right before their like absolute minimum? Do you still see that 1 critical ratio threshold?
Well, that was the 3rd chapter. I didn't subject you guys to that. Was to see at what point does the noise level below threshold start to impact hearing. It's about 10 dB below hearing is when you start to see the kind of influence of noise. But it's not until the noise hits the actual hearing threshold that the critical ratio takes hold, I guess. It's like in the same way that as noise bandwidth gradually widens to hit the critical band, the threshold will slowly increase until it gets to that critical ratio point. Same with level. It will gradually increase until it locks in, and then that critical ratio holds as you go up.
But in theory, then, a noise that they can't even perceive is affecting them.
We also measured that, which I just, also added in there. And it's the order of operations is that the noise is audible but not impacting hearing, then it starts to impact hearing, and then the critical ratio takes place. Yeah, and we just did the same kind of hearing measurements. Instead of listening for a pure tone, they were listening for the noise stimulus.
Okay. Thank you, Ryan.
Thank you. Yeah, that was great.
I'm sorry, Dr. Jones.
Oh, yeah. Only he has to call me that. You guys don't have to call me that. My car doesn't work. Does this thing sometimes?